Thank you all for attending this very special time in the life of Miss Madeline Totten. This microphone is very loud. Okay, is that better? Still a little loud? I'll just hold it away. I've been trying to convince Madeline this week that thesis defenses are actually a time of celebration of success more than they are an examination of content knowledge. And so this is a very special time in the life of a university. Probably in the life of a graduate student, this event is more important than even commencement itself, because this is the time of celebrating all the progress she's made and the work she's done. Madeline will be moving on from Marion in the summer to attend New York Institute of Technology's College of Osteopathic Medicine in Jonesboro, Arkansas. She has also applied to their DO PhD combined program, and so she should hear in a couple of weeks about the PhD portion of that program. I wanted to read just very quickly some comments that I made about Madeline to that DO PhD admissions committee just a few weeks ago to tell you from my perspective why she's such a special student. Madeline joined my musculoskeletal research group in May 2018 with the goal of advancing 2 lines of work that had stalled due to significant technical challenges. With no prior experience in tissue culture or expression analyses, she successfully navigated the difficult task of optimizing lentiviral transduction and selection protocols for 2 different mammalian cell lines. Her work opened the door for both projects to advance toward publication, and Madeline is qualified for authorship on both. I've been deeply impressed with how quickly Madeline established herself as a valuable, trustworthy, and skilled junior collaborator. From our earliest meetings, I observed her ability to swiftly comprehend new information and synthesize themes. Indeed, I've been privy as Madeline formally and informally explained her thesis work to fellow medical students, graduate students, and undergraduates with an exceptional degree of mastery, particularly given her nascent career stage. Without doubt, Madeline embodies the qualities that will make her a successful physician scientist. And without further ado, Madeline, the floor is yours.
All right, is this on? Thank you, Dr. Lowry, for the introduction. I've had a really awesome experience with the thesis track of this master's degree, and I'm really excited to share my work with you. So we can go ahead and get started. So first I'll give some background information on bone as a tissue. So bone is a dynamic, specialized connective tissue with many important functions in the body, some of which being storing minerals such as phosphate and calcium, providing structure and support, protecting vital organs, allowing for locomotion, and housing bone marrow. Bone is categorized as either cortical or trabecular bone. Cortical bone, also called compact bone, makes up about 80% of the bone. It's very strong and rigid, and it can withstand a lot of stress. However, it does tend to be a little bit more brittle than trabecular bone. Trabecular bone is also known as spongy and cancellous bone, and it makes up the other 20% of bone. It's much less dense than cortical bone, and it has a spongy appearance, hence the nickname spongy bone. Bone tissue is composed of hydroxyapatite, water, type 1 collagen, and other non-collagenous proteins and proteoglycans. The overall strength of bone is influenced by multiple factors, including the balance between cortical and trabecular bone, the composition of organic and inorganic material, and the structural properties such as the overall bone geometry and distribution. Bone is continuously being broken down and reconstructed through the processes of osteogenesis and bone remodeling to remove old microdamaged bone and replace it with new healthy bone, as well as maintain mineral homeostasis. Bone remodeling occurs in 4 phases: activation, resorption, reversal, and formation. The remodeling process is mediated by osteoblasts, osteoclasts, and osteocytes, which you can see in the histological image in the top right. The process begins when osteoclasts, our precursors, proliferate, differentiate, and become mature osteoclasts. Osteoclasts are large multinucleated cells that originate from hematopoietic stem cells. Osteoclasts are recruited to the bone surface, where they secrete hydrogen ions and hydrolytic enzymes, which digest and break down old bone. Once they are done breaking down the bone, it leaves a resorption pit, and the osteoclasts cease activity and undergo apoptosis. Osteoblast precursors are then recruited to the resorption pit, where they differentiate into mature osteoblasts and adhere to the bone surface. There, they synthesize and secrete new bone matrix, which becomes the new bone. Osteoblasts can then remain as bone lining cells. They can undergo apoptosis, and they can become entrapped as in the bone matrix and differentiate into osteocytes, where they function as mechanosensors, responding to stress signals that the bone receives and regulating bone remodeling. Healthy bone requires a balance between bone formation by osteoblasts and bone resorption by osteoclasts. A loss of balance between the two can lead to many detrimental bone diseases. When osteoblasts outweigh osteoclasts, we see diseases such as osteopetrosis, sclerosiosis, and Van Buchem disease. When osteoclasts outweigh osteoblasts, we see diseases including osteoporosis. And I'm going to dive a little bit deeper into osteoporosis because that's the main disease that our lab researches. So osteoporosis literally means porous bone, and it occurs when the rate of bone resorption exceeds the rate of bone formation in osteoblasts. This leads to bone becoming very porous and brittle and an overall loss of bone mass. Osteoporotic bone deterioration often leads to fractures, with 1 in 3 women and 1 in 5 men over the age of 50 sustaining an osteoporotic fracture. Risk factors for osteoporosis include aging, hormone deficiency, particularly estrogen in postmenopausal women, lack of exercise, poor nutrition, smoking, drug or alcohol use, prior fractures, and a family history of osteoporosis. The current gold standard of diagnosing osteoporosis is measuring the bone mineral density using a dual energy X-ray absorptiometry, or a DEXA scan. The graph on the right shows osteoporotic fracture risk with varying femoral neck bone mineral density T-scores. So bone mineral densities with a T-score of less than or equal to 2.5 standard deviations below the bone mineral density of a young, healthy adult indicates that they have osteoporosis. You can see that the T-score range of osteoporosis, so the 2.5 or less, they have a much higher risk of osteoporotic fracture above the age of 50. Current pharmacological therapies for osteoporosis either aim to decrease bone resorption or stimulate new bone formation. Anti-resorptive agents, including bisphosphonates, decrease bone resorption and have been shown to reduce the incidence of fractures, but they can have long-term side effects. Anabolic therapies promote new bone formation, but they're very expensive and they can only be taken for 2 years. With the aging population increasing rapidly, the number of people suffering from osteoporosis is also expected to rise, as well as the cost associated with this disease. Studies predict that by 2025 in the United States alone, osteoporosis burden is expected to grow by about 50% to 3 million fractures and $25.3 billion in costs each year. Thus, development of an effective, affordable therapeutic treatment for osteoporosis is needed now more than ever. A better understanding of the molecular mechanisms involved in the functioning of bone-forming osteoblasts is necessary for the development of new anabolic osteoporosis therapies. So osteoblasts are derived from mesenchymal stem cells, which are multipotent cells that can differentiate into many different cell lineages. To differentiate into osteoblasts, MSCs receive signals from multiple signaling pathways, one of which being the BMP pathway. I'm going to dive a little deeper into osteogenesis and the BMP signaling pathway, as this is one of the main focuses in our lab. So the BMP, or bone morphogenetic protein signaling pathway, is involved in many processes in the body, including osteoblast differentiation, chondrocyte differentiation, skeletal development, cartilage formation, bone formation, fracture healing, and bone homeostasis. Thus, disruption of this signaling pathway is shown to cause many bone diseases. BMPs are members of the TGF-beta superfamily of ligands. Two distinct pathways of this superfamily are the BMP and activin signaling pathways. The BMP pathway promotes bone formation by stimulating the differentiation and enrichment of osteoblast progenitors. Activins typically have opposing effects to BMPs and promote bone resorption. So in the phylogenetic tree on the right, it's a tree of the TGF-beta superfamily. So you can see up in blue is the BMP subgroup. We also have a TGF-beta subgroup and an activin subgroup. The BMP subgroup signals through SMAD158, while the TGF-beta and activin subgroups signal through SMAD23. So BMP and activin ligands begin their signaling cascade by binding through heterometric receptor complexes involving type 1 and type 2 receptors. The ligands begin their cascade by binding to a type 2 transmembrane receptor serine threonine kinase. BMPs bind to two different type 2 receptors. They can bind to BMPR2 and ACVR2A and B, while activins predominantly bind to ACVR2A and B. After the ligand binds the type 2 receptor, the receptor recruits and phosphorylates a type 1 receptor, ALK236 for BMPs and ALK47 for activins. The phosphorylated type 1 receptor then initiates downstream signaling through intracellular SMAD proteins. With BMPs, they signal through SMAD158 and activins through SMAD23. The phosphorylated SMADs can form a complex with SMAD4, which then translocates to the nucleus and has varying effects on gene expression and transcription of target genes. Sometimes the effects of these pathways overlap and are redundant. However, studies have shown that there's antagonism that exists between the BMP and activin signaling pathways. There's many clinical and physiological conditions where the antagonism occurs, including the musculoskeletal system, pulmonary vasculature, tissue fibrosis, and cellular differentiation. However, the mechanism behind the antagonism between these pathways is mostly unknown. While BMPs promote bone formation, activins have been shown to promote osteoclastogenesis, inhibit osteoblastogenesis, and reduce osteoblast activity. We view these pathways interacting in a seesaw mechanism, in which one pathway increases at the expense of the other based on the strength of signaling that the cell is receiving. So as I showed previously, the ACVR2A and B type 2 receptors can be utilized by both BMP and activin ligands. Lowry et al. In 2015 have previously shown that if the type 2 BMP receptor, BMPR2, is removed from the osteoprogenitor lineage of the limb skeleton of mice, there's a decrease in activin signaling, a high bone mass phenotype, and an increased bone formation rate, while the BMP signaling levels are preserved. So the Western blot on the right shows that the level of BMP signaling measured by phosphorylated SMAD158 does not change, so it's preserved. But when we knock out the BMP2 receptor, we see that there's a decrease in the amount of activin signaling that's occurring, measured by the level of phosphorylated SMAD23. Studies show that activins compete with BMPs for binding to these receptors, ACVR2A and B, that they can both utilize, and that this competition could be a mechanism that underlies the antagonism that we see between these two pathways. A better understanding of the endogenous mechanisms regulating these pathways could yield new therapeutic targets for treating osteoporosis. Preliminary data for this project examined a model of competition between BMPs and activins based on ligand or receptor availability. We used W2017 cells, which are murine bone marrow stromal cells. These cells are a good model for our project because they're undifferentiated cells that can be used to study cell signaling events in osteoblast differentiation. So for each experiment, our cells were plated in 10% DMEM overnight. Once adhered, they were serum starved for 24 hours. Serum starving ensured that the results that we saw were due to endogenous signaling in an autocrine or pericrine manner. After 24 hours, cells were either collected for analysis or they were treated with varying antagonists for 4 hours and then collected. First, we examined the effects of sequestering activin ligands before they're able to bind to their type 2 receptor. We did this using follistatin, which is an activin binding protein. So it binds to these activin ligands extracellularly and neutralizes their effects and does not allow them to start their signaling cascade. We hypothesized that if the activin ligands were sequestered, we would see an increase in BMP signaling due to the mechanism, the seesaw mechanism that we see between these two pathways. And this result is shown in the Western blot on the right by the level of phosphorylated SMAD158 to total SMAD1. This led us to believe that under endogenous circumstances, activin is somehow repressing BMP signaling, which is why when we sequester activin ligands, we see the increase in BMP signaling measured by the phosphorylated SMAD158. Next, we wanted to see if this also happens on the opposite side of the seesaw. Cells were treated with the natural extracellular BMP antagonist called noggin, which functions to sequester the BMP ligands before they can bind to their type 2 receptor. We predicted that similarly to treating with follistatin, due to the seesaw mechanism of interaction between these pathways, we would see an increase in the activin signaling that's occurring. However, we saw that there was no change in the amount of phosphorylated SMAD23. And we knew this wasn't a technical error because we did see that noggin caused a complete block in BMP signaling from occurring. Since follistatin and noggin both inactivate ligands upstream of receptor engagement, we were interested in blocking the signal transduction pathway further downstream to examine the effects of activation of the signaling cascades. Cells were treated with SB431542, which is a small molecule inhibitor of the activin type 1 receptors ALK4 and 7. Interestingly, unlike the increase in BMP signaling we saw when we treated the activin ligands with follistatin, SB431542 had no effect on the amount of BMP signaling that is occurring. And we knew this wasn't a technical error because this drug did stop activin from signaling through its cascade. So to summarize our preliminary data, BMPs and activins operate in a seesaw manner with the relative weight of one side of the seesaw determining how the osteoblast responds to the signals it's receiving. Follistatin sequesters ligands extracellularly and causes an increase in the activation of the BMP signaling pathway. Noggin sequesters BMP ligands extracellularly and inhibits the BMP signaling pathway, but it has no effect on the activation of the activin signaling pathway. And SB431542 blocks the activin type 1 receptors and stops activin from signaling, but it has no effect on the activation of the BMP pathway. This is where I picked up and began driving this project. Our preliminary data led us to hypothesize that the activin-mediated repression of BMP signaling in W2017 cells is ligand-dependent, occurs upstream of effector engagement, and is due to insufficient type 2 receptor availability, causing activin and BMPs to compete for receptor engagement. We decided that the best method to test this possibility was to flood the system with excess ACVR2A. So this approach required delivering cDNA and coding ACVR2A to our W2017 cells. Prior work in the Lowry lab involving lipid-based transient transfection in these cells was unsuccessful. I initiated a new line of experiments using transduction with 2 different serotypes of an adeno-associated virus. This was also unsuccessful, possibly due to low tropism for this cell type, and this was the first major roadblock of my thesis work. Fortunately, another project that I was working on had success using transduction with a lentivirus in a different cell line, so we decided to try a lentivirus in our W2017 cells. So transduction is a viral delivery system in which cells are treated with a virus encoding a gene of interest, and the virus integrates itself into the host genome. We first used a lentivirus encoding GFP, or green fluorescent protein, to optimize the transduction protocol in the W2017 cells. We used microscopy and quantified the fluorescence of our treated cells to confirm transduction. And this worked, as you can see by the picture in the bottom left. The green cells are the cells that were transduced with the virus carrying GFP. We then transduced our cell line with a lentivirus carrying cDNA for ACVR2A with a C-terminal V5 epitope tag. Unfortunately, before we were able to collect these cells and use them for experiments, they became contaminated, and I reached my second roadblock of my thesis project. So we repeated the GFP and the ACVR2A transduction protocols, began adding 1X antibiotic antimycotic solution to our media, and we hoped for the best. Thankfully, we were able to establish stable cell lines with both of these treated cells, and we performed Western blots to verify the transduction and overexpression. The Western blot on your left shows a protein levels with a V5 antibody. This tells us that the cells were successfully transduced. If you recall, the lentivirus with the ACVR2A had a V5 epitope tag, while the GFP virus did not. The Western blot on the right shows protein levels with an ACVR2A antibody. So this tells us that compared to the control, the cells with the ACVR2A lentivirus significantly overexpressed this receptor. And when we measured this quantitatively, it was about 700% over the control. We used the antibiotic pyrimycin to select for the GFP and ACVR2A treated cells. Both of the lentiviruses carried pyrimycin resistance, so treating with pyrimycin would ensure that any of the cells that had not been transduced would be killed, and the cells that were transduced would remain viable. We then wanted to repeat the follistatin treatment to examine the effect of BMP pathway activation in the cells overexpressing the ACVR2A receptor. We predicted that if the shared receptor was the limiting factor for these pathways competing against one another, flooding the system with excess receptor would therefore eliminate the competition, and follistatin would not increase BMP signaling as it did normally. We saw that the basal phosphosmad1 signaling was up, and that follistatin did not increase basal signaling any further. We also saw that treating the ACVR2A overexpressed cells with follistatin did not lead to an increase in BMP signaling. So in our GFP control cells, we saw that same increase in BMP signaling, but when we had the ACVR2A receptor overexpressed, it did not change the amount of BMP signaling that was occurring. This provided mechanistic evidence that the activin-mediated repression of BMP signaling occurs at the level of competition for receptor utilization. We were thrilled to get this result a few weeks before leaving to present this work at the ASBMR conference in Montreal. Unfortunately, I hit another roadblock when trying to repeat this experiment for statistical analysis. My Western blots were not working, and it took a few months and a lot of trial and error before we finally tried a new type of gels and were able to duplicate the results of this experiment. So to conclude, BMPs and activins interact in a seesaw mechanism with one pathway increasing at the expense of the other. BMPs and activins compete for the shared type 2 receptors, ACVR2A and ACVR2B. And finally, although it took a few roadblocks to get here, we can conclude that activins endogenously repress BMP signaling in osteogenic W2017 cells due to limited availability of type 2 receptor. At this point, we came up with a few future directions of where we want to take this project next. One we plan to take is examining the functional effect of activin-mediated repression on BMP signaling in osteoprogenitor cells. A second direction is sequestering activin ligands using neutralizing antibodies. And third, identifying other strategies of increasing BMP signaling. First, I will outline our plan for examining the functional effect of activin-mediated repression on BMP signaling. So our findings thus far have evaluated short-term sequestration of activin ligands, and they do not provide information on the functional effects of repression with regard to osteoblast differentiation and activity. So I propose to treat W2017 cells with follistatin for 24 hours and examine the effect on expression of the osteochondral marker RUNX2 and osteoblastic markers osterix, osteocalcin, and sclerostin by RT-PCR. I will also examine osteoblast differentiation and activity using alkaline phosphatase staining and osteoblast activity with alizarin red staining. I predict that follistatin treatment will lead to increased osteoblast differentiation and activity in these cells. These results would provide evidence that under endogenous conditions, activin ligands exert autocrine and or paracrine effects to limit osteoblastic differentiation of the W2017 cells. We will also run a series of co-treatment experiments using follistatin in combination with the BMP pathway inhibitor LDN-198189 to confirm that the effects of activin on blocking osteoblast differentiation are due to repressing BMP signaling. The second future direction is sequestering activin ligands using neutralizing antibodies. The goal of this future direction is to find a strategy to alter the ratio between BMP and activin signaling pathways. One approach to changing this ratio has been treating with BMPs clinically to induce osteogenesis. While effective for bone formation, treatment with BMPs has been found to have very harmful side effects and complications, including inflammation, swelling, ectopic bone formation, osteoclast activation, and increased adipogenesis, likely due to the large quantity of BMP ligand required clinically. Thus, a better approach is needed to reduce activin signaling. Our goal is to be able to reduce the activin signaling and allow the osteoblast lineage to interpret this decrease in activin signaling as an increase in BMP signaling. We plan to achieve this using anti-activin neutralizing antibodies. Current strategies for blocking activin signaling in vivo are nonspecific and may have potentially harmful side effects. Using a neutralizing antibody that targets an activin dimer is a more specific approach that could potentially be used to increase bone mass. I propose a study in which cells will be treated with neutralizing antibodies against activin A or B ligands to investigate if neutralizing antibodies can initiate an increase in BMP signaling. If this hypothesis holds true, I propose an in vivo study examining the effects of anti-activin antibodies on bone mass in mice by systemic injection. I predict that treating with a neutralizing antibody in vivo will lead to an increase in bone formation and bone mass. So far, we have confirmed that the W2017 cells express both activin A and B ligands using RT-PCR. So next, we plan to treat the cells with neutralizing antibodies against these ligands, and we predict that treating them will lead to alleviation of the activin-mediated repression of BMP signaling. Rationale for our hypothesis that we will see an increase in BMP signaling in vitro and an increase in bone formation in vivo when treating with neutralizing antibodies comes from multiple studies. Chantry et al. Showed that in normal conditions, treating with an activin ligand suppresses bone mineralization. So this image on the right is alizarin red staining, which measures the bone. It's used to quantitatively detect bone mineralization by staining calcium phosphate crystals. So when they treated with an activin A ligand in both circumstances, there's a significant decrease in the amount of staining occurring, telling us that there's a lot less bone mineralization occurring. However, when they treated with either ACVR2A protein and the ligand or ACVR2A protein alone and a monoclonal antibody against activin A with the ligand or the monoclonal antibody alone, they saw an increase in bone mineralization. Hatzel et al. Showed that using an anti-activin A antibody in a mouse model of the disease fibrodysplasia ossificans progressiva, also called FOP, blocks progressive heterotopic ossification. So that is when skeletal muscle is abnormally converted into misplaced bone. So this image on the bottom left shows their mouse model of an FOP mouse, where they have these bone lesions form abnormally. However, when they treated with an anti-activin antibody, we do not see the same heterotopic ossification occurring. This antibody is currently being used in a clinical trial for humans with FOP to examine the safety and tolerability of administration of this antibody in humans. Our third future direction is identifying other strategies of increasing BMP signaling. If the outcome of the clinical trial with the anti-activin neutralizing antibody indicates unacceptable adverse event rates in humans, an alternative approach to the neutralizing antibody strategy is using an activin-based propeptide inhibitor. So McKandry et al. Designed this propeptide by linking residues of an activin A propeptide to a TGF-beta1 propeptide. Treating with this activin-based propeptide specifically antagonized activin signaling. Another approach would be targeting this antibody to bone using bisphosphonate conjugation. If you recall, Lowry et al. Showed that knocking out the BMPR2 receptor in vivo led to an increase in bone mass. I have shown in the Western blot over on the right that treating W2017 cells with a neutralizing antibody against BMPR2 leads to an increase in BMP signaling measured by phosphorylated SMAD158. Thus, I would predict that treating with an anti-BMPR2 antibody in vivo would lead to an increase in bone mass. I'd like to thank my research mentor, Dr. Lowry, my thesis committee members, Dr. Plotkin and Dr. Hum, my lab partner, Kelly Justice, the funding that made this work possible, the Bone Mineral Research Group, and Marian University. And I will take any questions that you may have.
So we can now transition to the public questioning, and then we will have the private questioning of Madeline for a thesis following this.
So backing up to when activin signals through ACVRA/B receptors, why did you choose to upregulate A instead of B?
Great question. So they can signal through both ACVR2A and B, and we knew we wanted to overexpress one of them, so we kind of just picked one. ACVR2A and B have about an 80% homology between the two, and their ligand binding domains are mostly identical. There's like barely any difference between the two. So we would predict if we were to repeat these experiments overexpressing ACVR2A or ACVR2B, we would see the same results. Yes?
I have a couple questions. The first one is your I'll have to read it right here. ACVR2A and B, have their structures been solved?
What was the second part?
Have their structures been solved?
I believe so. And they're just very similar. There were a few minute changes between the two, but overall the binding sites are similar.
So the ligand binds to a common binding site?
I'm not sure if I know the answer to that. I think it picks one of the two. Like it can bind to either one with similar affinity, and they're so similar that it still has the same signaling effects downstream, whether it binds to the A or B version.
But the ligand causes a different SMAD response, even though it hits the same binding pocket.
Correct. So like whether BMP or activin binds to it, that's correct. And that's just because in that image where the ACVR2A and B receptors are in the middle, we kind of view it as they can pick the activin signaling cascade, or they can go to the BMP signaling cascade, and it just depends on which ligand is binding. So if BMPs bind to the binding site, then we see the BMP signaling compared to the activin signaling.
So are there certain different partner proteins or other things in between the receptor and when you get that transduced down to the SMAD for which particular way you're going? Because you'd think there would have to be a reason why you could stimulate the same binding pocket and get different result.
I think, I'm not totally sure, but I think that depending on which ligand binds, that's how the type 2 receptor knows which type 1 receptor to recruit. So with the BMP ligands, it recruits the ALK 236 type 1 receptors, and then that starts the signaling cascade. And when activin binds, it recruits the ALK 47 type 1 receptors. There's probably more to it than that, but that's as much as I know about it. That's right. Yes.
Follistatin inhibits activin?
Correct.
What's the difference between, then why do the neutralizing antibody? Don't you already have the reagent that you need there?
For the neutralizing antibody?
Well, no, the neutralizing antibody also inhibits activin, right?
Yes.
So what's the difference?
So follistatin is not, does not only target bone. It has effects in the whole body. So it would not be a good model for potential treatment in vivo. Whereas with the neutralizing antibody, those other studies have shown that you are able to target bone. So that's why we would hope we would still see the same results treating with the neutralizing antibodies. Did that answer it?
So this is new to me, so I'm trying to think this through. So that means that there's a different targets for follistatin, but you can create a monoclonal antibody that's specific only to the activin that activates bone?
Yes. So whereas follistatin would sequester activin ligands everywhere that we see the activin and BMP signaling cascades occurring, hopefully a neutralizing antibody to those ligands however we delivered it to bone, it would only sequester those activin ligands in bone and not elsewhere. Hopefully. Any other questions? Thank you all for coming.